Michael, why don't you take it away and tell
Right.
us a little bit more about Jen?
Sure. Well, actually, instead of telling us about Jen, I was going to ask us about Jen a little bit more, get her to tell us a little bit. And I know, Jen, there's so many cool things, but one of the coolest things that we first heard from you was about this portable gene therapy, or as the people in Seattle, the community members around me would call it, gene therapy in a box. And I don't know if you wanted to start off by telling us a little bit about your work with that, and then maybe go into some GGTI. How would you like to do this?
Sure. So I can say, a big passion of mine has always been how do we bring transformative therapies to the people who need them? I will say I've not always been in the HIV space, but the HIV space changed my trajectory along my career when it presented a pretty big problem to me, which was how do we take one of these sort of boutique therapies and actually make it accessible and affordable and sustainable for a disease impacting more than 37 million people around the globe, most of whom do not live in Seattle, Washington, where I am from. And so that is really a lot of the inspiration for the work that I do and for the Global Gene Therapy Initiative. In the talk I'll give today, you will get to hear about why gene therapy is important and what is happening in the field right now. That means it's more important than ever for us to be thinking about how we're going to make this work for people around the world.
Yeah, it's interesting when I heard you say boutique therapy. It really does, I think, hit the head of the nail right on the right where it counts, because it is sort of a boutique therapy. It is something that only a few people who are select will be able to get to experience. And yet we know that it's so important because it's really curing or helping to eliminate the conditions and the diseases that people suffer from all around the world. So to take it from boutique to commonplace is definitely going to be the challenge that I know you and I both feel very strongly about. So did you want to start off with sharing your own slides?
Sure.
All right, let me do this. I'm going to remove the spotlight here. There we go. There you go. And if you want to,
yeah, how's that look?
It's looking good.
Perfect. So I just wanted to briefly start with a land acknowledgement to the original caretakers, in particular of the land that I sit on, the Duwamish, Tulalip, Muckleshoot, and Suquamish tribal nations. Without their stewardship of this land, I would not be sitting here doing the work that I do and being able to share that passion with all of you.
Awesome.
Let's see how we advance here. So as the flyer promised, today is the basics, an introduction to gene therapy for the treatment of HIV. And we have quite a bit to cover. I thought I could look at all of the different lectures you're about to see over the next few months and give you kind of the best 30,000-foot view to prepare you for these different approaches and what they could mean for the field. Mm-hmm. So what are the basics? What I hope you're going to walk away from today is an understanding of what gene therapy is and why you should care, how gene therapy for HIV works, and how far we've come up to date with gene therapy to treat HIV and the lessons we've learned so far. And then, as Michael and Linda mentioned, I'm hoping to get through this in enough time for you to ask questions. Please feel free at any time to put questions in the chat. And at the end, I'll have a few extra resources to put in there as well. So let's roll right into it. What is gene therapy and why should you care? Now, to explain what gene therapy is, I have to review some basic biology of living cells. This is just a super simple diagram of any living cell, whether it's a cell in a plant or a human. But for the purposes of today, we'll focus on human cells. Cells have a complete copy of the blueprint for whatever living thing they are part of in a compartment of the cell called the nucleus. And this blueprint is a molecule we call DNA. These are the complete instructions for all of us. Every cell has that entire blueprint. But there are different types of cells and tissues in our body. We have liver cells, skin cells, eye cells, toenails, hair, heart cells. Each of those cells needs to serve a different function. So each of those cells is only using the section of the blueprint that applies to them. So heart cells are only using the heart section of the blueprint, skin cells only using the skin section of the blueprint, and so on and so forth. In every cell, there are little DNA readers, proteins that scan the DNA and the sections that in particular pertain to that cell or tissue, and they read those instructions, which we call genes. When a cell receives kind of a message from the environment that it needs to do something, it will read the directions, read the blueprint for that section, and it will produce what I like to call an intracellular memo, or a molecule called RNA. Now, for example, if this cell were one of my pancreatic islet cells, it might have received a message from my body saying, Jen ate a whole bunch of cake today, and we have way too much sugar circulating in our blood, and she needs more insulin pronto. The DNA reader would find the instruction in the blueprint to make insulin. It would write the memo saying, insulin needed urgently, here's what it needs to look like. That memo would go to a different part of the cell called the cytoplasm. This is really the factory where this insulin is going to get made. It will get assembled into a protein. And then in the case of insulin, it gets secreted out of the cell into the bloodstream so that it can help manage my blood sugar levels. This is just one example of one gene in one kind of cell. Now imagine that your body is full of billions of cells carrying out hundreds or tens of thousands of genes worth of functions in different priorities based on the cues you get, not just from what's in your blueprint, your genetics, but also the things that you're exposed to in your environment. And this is called the central dogma of biology. The blueprint is in our DNA that gets transcribed into memos, RNA, that get translated into proteins, which result in cellular structures and functions that make us alive. So when we talk about gene therapy, what we're really talking about is manipulating or modifying the genes so that we can alter the biologic property of the cell for a therapeutic purpose. And this is the actual US Food and Drug Administration definition. So you notice I highlighted and underlined the word alter here. I want to take a minute to talk about kind of the flavors of alteration that we could think about in the context of this biology. So the first is turning off disease-causing genes. Some people are unfortunately born with genes that cause diseases. Others have changes to their genes made by things they're exposed to in the environment that can then cause disease later on in their life. A very simple flavor of gene therapy is to turn off a gene that is causing a disease to render a cell healthy. A second type is replacing a gene with a mistake in it with a healthy copy. So sometimes we can't just turn a gene off because we actually need that memo and that protein in order to survive. So in this case, we can actually try to replace the gene that has the mistake in it with a working copy. And the last flavor I want to talk about is adding something new, creating an addendum to the blueprint, if you will. This would be taking the code for a gene and putting it into our DNA, even though that instruction was not ever part of the original blueprint. And this is something that is done a lot in T cells. We're going to talk a little bit about this later on in the presentation. In particular for HIV gene therapy, turning off disease-causing genes and adding new genes with disease-treating properties are the two most common flavors that get applied. And we'll talk about how that's done over the course of the webinar. So a lot of people ask, why should we care about this? This seems really Mystery Science Theater 3000. You're going after my blueprint. Why would you want to change my blueprint when I can just take antiretroviral therapy and keep my HIV infection suppressed? Why would we need something as out of the box as gene therapy for the treatment of HIV? Well, not everyone on the planet has access to antiretroviral therapy. And if we can't get enough people on antiretroviral therapy whose HIV is suppressed, then we continue to get new infections. every year. Some people can't take antiretroviral therapy due to side effects, and it's not a cure. It has to be taken reliably for the rest of the life of the person living with HIV, or you can get a resistant HIV strain developing. And you can also have potential other complications from the HIV, which we'll also talk about in the next section of this presentation. So this is what I was just talking about. UNAIDS put forward these targets, the number of people that would need to know their status, know whether or not they're HIV positive, how many of them would have to be on antiretroviral drugs, and how many of those who knew their status and were on antiretroviral drugs would have to have their virus suppressed in order for us to be able to control the HIV infection across the globe. And the yellow line kind of shows you where we are up to 2020. The orange dot shows you the target they were hoping we would be at by 2020 to control infections. And what I hope you can see from this graph is that we didn't meet it.
Mm-hmm.
The other problem is the amount of money it takes to meet those goals. Unfortunately, funding for a lot of these programs has stagnated, which means that we're not getting increases. And as the case numbers stay higher, you actually need to expand those budgets to meet those criteria. And we're not meeting those as well. So you could see the target in the upper right-hand corner of this graph and the amount of funding and the places that it's coming from shown in the bars up through 2018. And unfortunately, we did not have a great spike in 2019 and 2020, so we did not meet those funding goals either. This means that we need to think a little bit more creatively about how we're going to manage this moving forward. And gene therapies have a lot of unique properties that I'll talk about on this slide. So first of all, because we're actually going at the blueprint or the basic biology of the cell, gene therapies can potentially be curative or can go after the underlying disease biology rather than just managing the symptoms of the particular disease or infection. This means that they could be a one-time treatment with curative potential. So imagine a single intervention where all of a sudden virus is suppressed for the rest of a person's life without the need to take antiretroviral drugs. That would be something that would be really transformative, and it's the ultimate goal of approaches like gene therapies. We do have gene therapies that are approved by the US Food and Drug Administration and European Medicines Agency, 10 so far and 2 more that just were recently scheduled for approval. And right now in the United States, the Food and Drug Administration receives about 900 new applications per year for some type of gene therapy to treat some kind of disease, and more than, and I think it's now we're up to something like 189 different diseases that are being investigated as potential diseases where gene therapy could be useful to treat.
Mm-hmm.
This means that the field is super fast paced, and that's bringing a lot of investment into it. In terms of biotechnology and commercialization, this is pretty record breaking, with about $2.7 billion US proposed to be invested by the year 2026. Mm. So for all of these reasons, gene therapy could be a valuable tool in the box or the suite of tools that we use to keep the HIV epidemic under control and limit the negative effects that it has on people living with HIV, no matter where they exist in the world. So I hope I've given you a good definition or introduction to what HIV or what gene therapy is and why it matters to the HIV space. Now I want to get a little bit more granular and talk about how gene therapy looks for HIV specifically. And to do that, we have to talk about what HIV does to our cells. And it's interesting because HIV actually takes advantage of that central dogma that I just told you about, where our DNA begets RNA, begets protein, begets function. HIV, as a particle in nature, which kind of as a cartoon looks like what's in the upper right-hand corner of this slide, it's not really doing much, but it actually contains its own blueprint, its own little set of virus genes, genes that if there were readers and memos and factories would make new virus particles, but none of those things exist in the particle by itself. This is why HIV wants to get into our cells, because we have readers and memos and factories that can make new viral particles.
Mhm.
So why doesn't HIV just go into any cell that has that? I already told you every single living cell has a copy of the blueprint, readers, factories, etc. Why is it selective? Well, this is probably part of its evolution, but it's a virus, right? And when we get infected with viruses, I'm getting over a cold right now, which is why I sound a little stuffy. Our immune system naturally is designed to say, whoa, 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 you are not part of Jennifer. I do not recognize that blueprint. You need to go, bye-bye. And so HIV sort of said, well, what if I selectively do my business in the immune cells? What if I take over those cells? Then the immune system maybe won't see me as much of a problem, or I'll take out the immune system that would normally take me out. And so HIV particles, they're looking for specific things on the surface of cells that call out immune cells. In this case, we call these proteins that are on the surface of cells receptors, and many of them have names that start with the letter CD. That just stands for cell differentiation. A long time ago, doctors, scientists looking at cells under a dish said, well, these cells look different from those cells, and I see this thing sticking out on the top of that cell that I don't see sticking out on the top of that cell. And so I'll call this one cell differentiation marker number 1, 2, 3, 4, whatever. There are actually hundreds of these things. So CD4 was actually the fourth one that was identified. That's why it's called CD4. CD4 probably was identified early because it's super big and bulky and kind of easy to see even with earlier microscopes. And HIV kind of says, okay, I'm going to use CD4 as sort of my gateway to the immune system. But CD4 by itself is not enough for HIV to get into the cell. It needs another molecule, and we call this a co-receptor because it has to also exist on the surface of the cell close to the CD4 molecule. And there are a couple of different co-receptors that HIV can use. The one that's most commonly used is called CCR5. And then there's another one that people talk about a lot called CXCR4. When you're listening to scientific talks about HIV, you might hear R5 and R4 tropic. That just means whether or not the strain of HIV prefers CD4 plus CCR5 or CD4 plus CXCR4, which is where those names came from. But particle gets in, it finds a cell that has both CD4 and let's say CCR5 for purposes of this talk today. Now it can actually get close to the cell surface and get inside. And this is where it does something super clever. It doesn't just start making new virus particles, it actually takes its viral genes and it permanently puts them into our blueprint, like an addendum. It says, we're going to just hide here in the blueprint as our own page of instructions so that when the DNA readers come along, they're going to think, this is an urgent memo. We need you to make a lot of these genes right away. It's totally important for bodily function. Please get it done quickly. This means that the cell will start to make lots of viral proteins, which in turn generates new HIV particles. So now you have an immune cell in the body that instead of reading its DNA and doing the things that immune cells are supposed to do, is now instead putting all of its energy and effort towards making new HIV viral particles. And it will make a lot of them. So much so that oftentimes the T cell doesn't survive because it's not sustaining itself, it's only sustaining the HIV. This means that new particles get released. A cell doesn't have to die to release new particles, but cells that do die release a lot of new viral particles, and you get the system perpetuated. Now you've got this many more particles that can infect new cells, and those cells can then become factories to make new HIV particles, and so on and so forth. So what does that look like? If we were monitoring a person clinically, it would look something like this. So you could see I have this vertical axis, which just is the amount measured in blood. And we're going to follow two things: the amount of virus particles or virus copies in the red line, and the number of T cells, the immune cells that HIV is going to preferentially go after in the blue line. The horizontal axis is how much time has passed after the initial infection. And the dashed line represents if we were measuring the virus in blood, that's kind of the golden zone. We want to be below that line. If we're below that line, the virus is considered undetectable in the person's blood, and that means that they can't transmit HIV to someone else. So what typically happens after infection is you see this number of virus copies go up as lots of T cells get infected and start making new virus copies, and some of them start to die. And so you start to see the T cell numbers go down. That's the blue line. But the virus is smart. It doesn't want to kill a person. It wants to make virus forever. It wants that person to become a factory to spread HIV so that it can perpetuate itself. So what it tries to do is sort of level out, say, here's just enough virus and just enough T cells for us to be in that happy medium without the person dying. But what the virus doesn't think about are other infections, right? So the problem is if you have lower T cells, you're susceptible to all sorts of other things that your immune system would normally try to be fighting off. And if you are susceptible to those infections, then you can have lots of complications and other problems that compromise your health. But it's typically in this space, sort of this, we call this the set point, the place where the virus and the T cells have sort of leveled out, which is when most people get diagnosed or get tested, and then they begin antiretroviral therapy or ART. This knocks down the virus levels. So we see that red line go below the dashed line, and it lets the T cells start to recover, sort of brings them back up. And if you stay on antiretroviral therapy without side effects and there are no other complications and you can maintain that, this is the pattern that can carry on for the rest of your life. But if for any reason you have to stop the antiretroviral therapy, we typically see this very quick rebound of the virus. The red comes right back up and the T cells go right back down. And this is really problematic. When we first started looking at antiretroviral therapy, people got really excited. It's keeping the virus super low. It's undetectable. Maybe, just maybe, we've given the immune system enough space and time that it can actually fight off the infection and then potentially cure the person itself. But unfortunately, when antiretroviral therapy was stopped and they saw these patterns, they realized that that was not the case. And that was when we learned an important lesson about HIV. It actually produces two kinds of infected cells inside the body. The cells that we talked about are those red cells on the left of this slide, the productively infected cells. These are those CD4 T cells that are making new virus particles. Their readers and their memos and their factories are just producing new HIV. But there's also this other kind of cell, the blue cell on the right side of the slide. And this is called a latently infected cell. Here, the HIV has hidden its genes, its little addendum to the blueprint, and the cell is not doing anything with them yet. It's actually harboring them like a fugitive after a bank robbery or something. They just get to sit in there, and the body is none the wiser because nothing suspicious is happening quite yet. These latently infected cells are the ones that cause that viral rebound. So the antiretroviral drugs are really designed to go after the productively infected cells, and they don't stop the latently infected cells from being able to make new virus later on. So which cells are latently infected? Well, lots of different kinds of cells. There are cells all over the body of lots of different types where these sort of latent infections have been found. And that means that HIV is very clever at hiding like a fugitive in lots of different places in order to evade antiretroviral therapy surveillance. This means that we have to think about approaches that can not just go after those cells that are actively producing HIV, but cells that are hiding HIV or harboring HIV genes. Now, there are some people that have been studied who actually do this on their own without the benefit of antiretroviral therapy. And these give us some clues as to how we could go after the biology of HIV infection with something like gene therapy to actually improve therapeutic outcomes. So many of you may have heard of people called elite controllers. These are people who have been exposed to HIV and they do actually get infected. We see viruses go up and T cells go down, but then they don't hit a set point or they don't plateau to that happy medium. Instead, their T cells just keep going back up and their virus just goes back down, and they don't need to take ART to do that. And it happens to stay that way for the rest of their lives. Studying these people, there's wonderful people who have given their bodies and allowed us to study them so that we can understand how this happened. We learned two valuable lessons. Generally, these elite controllers have super strong T cell responses to the HIV infection. That means that if we can give T cells kind of a super robust first response, maybe we can control the infection a little bit better. But the other thing we learned about these people is that their latent cells, the cells that are harboring HIV genes, they aren't very good at making new virus particles. So even in the absence of antiretroviral therapy, they just can't kick into gear. So studying those cells gives us two ideas of how we could approach HIV infection with gene therapy. Either give T cells a strong response or make sure that the latent cells aren't so great at producing new viral particles. Another group of people are actually resistant to HIV infection altogether. These are people who have been documented to have been exposed to HIV numerous times, but we just don't ever see those patterns that I showed you on the previous slides. They never get virus copies that are detectable and their T cells never come down. Studying these individuals has shown us that if HIV doesn't have that co-receptor, in particular CCR5, then those T cells cannot be infected with HIV and the cycle never even starts to begin with. And so these are all clues for how we might be able to use something like gene therapy to impact the biology of cells in people who are living with HIV who are unfortunately not elite controllers or naturally resistant. This CCR5 approach is actually what was responsible for the first now 4 people who are reported to be cured of HIV infections, including the Berlin, the London, the Dusseldorf, and now the New York patient, or the first woman who was treated with this. In each of these cases, this was not a gene therapy, it was a cell therapy. And in particular, it was a cell therapy known as bone marrow transplant. In this case, we actually took the person who had HIV and they were treated with a combination of chemotherapy and other treatments that essentially wiped out all of their blood cell production. Now, they can't survive without blood, so they needed some blood. The blood that was given to them that will become the blood for the rest of their lives came from people who had been lucky enough to be born with a mutation in their CCR5 gene, and specifically the mutation in the CCR5 gene that is shown to be responsible for natural resistance to HIV infection. So of course, this sounds awesome. If we can replace the blood with resistant blood from a resistant person, that could work, but there's a big problem. When we do these kinds of treatments, you can't just choose blood from anyone. You have to choose blood from a person who is a close match, because the immune system is going to recognize what is not self. And if we put in an immune system that is not sort of looking similar, then it's just going to attack all of the tissues in the body. The number of donors, possible people who were born with the Delta 32 CCR5 mutation, is not even enough to treat 1% of all people living with HIV. That is why, while it will work for the lucky few who match to someone who is naturally resistant, it's not going to work for everyone, and we need a different approach. When we do gene therapy, as you'll hear about in the next few slides, we're actually using the patient's own cells. The cells are always the self. So the idea there would be, for example, to recreate the Delta 30 mutation. That's actually turning off a gene, right? That's one of the flavors of gene therapy we could do. So what if we just turned off the gene in the patient's own blood cells? Would that be enough to get HIV resistance or HIV control? Now, based on all of that biology, there's actually an incredible number of strategies that people are trying. Knocking out CCR5 is just one of them. And there are more. I could spend actually probably the entire series of this webinar explaining each one of these approaches. So for purposes of the basics, I'm just going to tell you that there are more approaches than I can count, and they generally fall into three categories. Defensive, that's preventing infection of new cells. It's very much like in the HIV resistant patient, like the CCR5 knockout, where you don't allow infection to occur at all. There's offensive strategies shown in the upper right. This is killing off the infected cells, and it's very akin to what the elite controllers do. A lot of times this is engineering the T cells to have that super active response against the HIV infection so that it wipes out the infection before it has a chance to set up house. And then the third category at the bottom, targeted HIV gene therapy. This is shutting down production of new HIV particles, and it's very much like that other condition of the elite controllers, where you try to make the latent cells not so great at making new virus. And all of these are reviewed in a paper that I'm going to show again in a couple of slides. It's a little bit science heavy. It's going to use a lot of technical terms, but I will put it in the chat at the end of the talk so that anyone can read it who would like to. So an important facet of all of these gene therapies is that the only way for us to know whether or not they're working is for a patient to not be on antiretroviral therapy and for us to see what happens to both the T cells and the virus. And this is something that a lot of people think twice about when they think about HIV gene therapy trials. And we're going to talk about more ways that we're trying to approach this moving forward. But it is a critical part. We can't know if the gene therapy is working unless we take away the benefit of the art and see what the gene therapy can do. And so this is an important consideration in all of these gene therapy trials. Now, I mentioned there were a lot. We're going to talk about a few of them and what patients had to go through. And so I just want to say to anyone who has ever participated in a clinical trial of any kind, I super appreciate your participation in science and scientific process. It is the only way that we learn things, but it does mean a personal choice on wanting more to contribute to that knowledge being gained than necessarily knowing that there may be a benefit to yourself. And this brings me to that C word that I mentioned earlier, curative. When we talk about HIV, there are really two forms of HIV cure. And it's important to introduce these because we have to think about them in the context of any new gene therapy. So the first is called eradicating. This would mean getting rid of both the red and the blue cells, getting rid of any cell that has any potential to ever make HIV, both the producing cells and the latent cells. And this would be akin to those HIV resistant humans that just never have an infection possible. The second is called functional cure. This is simply the same benefit that you get from successful antiretroviral therapy, but without the need for antiretroviral therapy. So your virus is undetectable for the rest of your life without the need to take antiretroviral drugs. And this would be like elite controller humans. It doesn't mean you can't get infected. It just means that you'll always be able to control that infection. So we've talked about what gene therapy is and why you should care. We've talked about what HIV gene therapy looks like and how it works. So how far have we come with gene therapy specifically to treat HIV, and what have we learned so far? Well, since 1994, there have been over 36 clinical trials using some kind of gene therapy to treat HIV infection that have been registered in the international registries. And this is that paper that I mentioned before, which summarized the different categories and which I'll put in the chat at the end, which you can read. But you can also look at the registry itself. It's www.clinicaltrials.gov at the bottom of this. Now, if you go to that paper where it lists the clinical trials, it lists a number, an NCT number. That just stands for National Clinical Trials Registry number. So you can go to that website and actually plug in the number for a specific clinical trial and see what that trial is about. But you can also go to that clinicaltrials.gov website and you could just search for HIV and gene therapy, and it will bring up trials that include those two words in them. And you can look at the individual trials. You can see where they're being conducted, who is running them, should have basic information on the approach that's being used. And oftentimes the trials have to update that registry with any progress. So if they publish a paper or if they announce clinical trial results, they have to include it in that website. And so these are tools that you can use to explore on your own time and in your own pace what is happening in clinical trials for gene therapy so far. There is one person that very recently, just at the most recent CROI meeting, was reported as possibly being functionally cured of HIV using gene therapy approaches. And we're calling this patient for now the Philadelphia patient. It's still very early and there's a lot of work to be done, but what is important here is that the patient just didn't get one treatment. They actually got 3 different interventions. That was not the intention at the start. This happened to be a patient who wanted to go forward with gene therapy. The first gene therapy didn't appear to work. When they came off their antiretroviral drugs, the virus came back, their T cells came down, so they went back on antiretroviral drugs. They got a second intervention as part of another treatment and clinical trial, and they came off their ART and the same thing happened. The virus went up, the T cells came down, and then they got a third intervention. And the first 2 interventions were really trying to introduce that mutation in CCR5, so shutting off the CCR5 gene. So the first 2, both times, tried to shut off CCR5. The third intervention was actually another intervention where they added a gene to the T cells to try and make them super active against the HIV infection. Now, what was interesting was after that third treatment with the T cells, when the patient came off of antiretroviral drugs, they saw a little bit of virus come up and a little bit of T cells go down, but then they quickly saw T cells come back and virus go away without having to take the antiretroviral drugs. But when they looked at the blood of that patient after this had happened, they saw that the CCR5 gene turned off cells were also there. Even though it didn't appear that they worked the first few times, it seemed like this combination of the T cells and the CCR5 knocked out cells was necessary in that third round for this patient to have the response that they did. Now, it's just one patient and it's still very early. And this was obviously not ideal. It was a lot of treatments over multiple trials, but we can learn something from this patient and we can think about what it might be telling us about how gene therapy using a patient's own cells could work moving forward. This was just to show you that this patient got both of those turning off and adding a new gene approaches. In particular, the new gene that was added was called a chimeric antigen receptor, or a CAR gene. I'm not going to go into too much detail because you're going to see a couple of webinars in this series that are going to go into more detail about CAR gene therapy in T cells and how that can boost T cell activity against HIV infection. So what did we learn from all of the studies that have happened so far, not just the Philadelphia patient? Well, we've learned that you need to get a lot of cells to have the gene therapy in order to have really good, robust activity against the HIV, to not leave it any room to change its behavior in a way that gets around the gene therapy. We also think that the gene therapy might need to include multiple strategies. We might need offensive and defensive, or defensive and targeted, or offensive, defensive, and targeted in order to get a really good response for a patient. But we also learned that gene therapy of blood cells to treat HIV can be safe and effective in such a way that a patient could receive 3 different treatments and still be doing okay. But each of these treatments is really expensive. And that has to do with how the treatment is done. So when we do gene therapy of blood cells, which is what was done in the Philadelphia patient and what would mimic what was done in the Berlin, the London, the Dusseldorf, and the New York patients, we don't necessarily want to put the gene therapy inside the body because we don't know what would happen if it got into blueprints for other cell types. So instead, we have the patient come to the clinic and we take the blood out of the patient. We then have to prepare the blood to receive the gene therapy. If we just want the gene therapy to go into T cells, we need to get the T cells separated from all the other cell types, for example. Then we have to add the gene therapy. There's multiple ways. I could spend an entire webinar series telling you about all the different tools that are available to get the gene therapy inside the cells. Each flavor can be done in a different way. And that's a process that's done typically outside the body. Then we have to actually look at the cells and make sure that they're okay. We've done a lot to them outside of the body. We want to make sure that they're not contaminated with something that could hurt the patient. We want to make sure that enough of them got the gene therapy that this could potentially be useful. Otherwise, why would we risk the patient's health by putting them back in? And we need to prepare the patient. So cells that stayed inside the patient's body the whole time are always going to have an advantage over cells that we did a whole bunch of stuff to outside the body. And so we need to make a little room for those cells coming in to ensure that they have a fair chance at actually accomplishing what we hope they would accomplish. And this is a complicated process that has to be done in super sophisticated facilities. This picture is from our facility inside Fred Hutch, where we do phase 1 clinical trials of these kinds of gene therapies. So it's not even the most sophisticated facility, which is typically the kind that is being used at a company to make these kinds of products. And you could see multiple pieces of equipment, lots of highly trained staff, lots of note-taking, people watching to make sure what was done, should have been done, et cetera. This kind of infrastructure is partially what leads to the super high price tags on these gene therapies. In addition, the first companies to get their gene therapies approved, they had to basically carve the path through this tall grass, right? Nobody knew what would be the right safety to balance with the right benefit, and what things should we be measuring and looking at? Obviously, in HIV, you would look at T cells and you would look at viral loads, but what else do we need? Do we need to look at, the person's quality of life? Do we need to look at other factors, liver toxicity, other things that we maybe necessarily wouldn't think about? And because they had to do all of those things to find the way that we could get to a balance of risk to benefit, they had to spend a lot more money than most companies that come after them will. And to balance that, they have hefty price tags for the initial drugs that were approved because they were the ones who had to carve that path. That said, it's not sustainable to keep these price tags the way that they are. And in particular, when we think about HIV, as I mentioned at the beginning, when Michael asked me that question, it was being asked to develop a gene therapy for HIV patients that made me think about this problem. So I was the person in that gown in that special room doing all of the things. And I kept thinking to myself, if we even benefit one person, how are we going to make this work for everyone? Because this is what the landscape of HIV infection across the globe looks like. The vast majority of people living with HIV live in South Africa, sub-Saharan Africa as a continent. And the cost of living and the cost of treatments there is not sustainable for the price tags on gene therapies that we have in this country. And then I looked at how many people were trying to do cell and gene therapy trials for HIV in Africa. And it was just as disappointing as this slide makes it out to be. So we have a disease that there is a standard case patient, which means a patient living in sub-Saharan Africa. And we're developing these really amazing next generation treatments, and none of them are being evaluated in the patient that's considered the global standard case. And so personally, for me and for many of the folks who are on this call that you heard speaking at the beginning, and you're going to hear, I hope, in several different venues, over the course of your journey through this field of research and space came together and said, we need to do something about this. And we formed what is called the Global Gene Therapy Initiative, or GGTI. Thanks to Michael Luella, who designed our logo for this. And this is really just a group of people who are passionate about the fact that we ought to be developing these therapies in the standard case patient globally. And it shouldn't necessarily be me, it should be an African for an African. We recently published a paper and highlighted that there are 4 things that need to be happening simultaneously for us to be able to do this. First and foremost, community outreach and education, which is why all of you here, I hope, are enjoying this lecture and why I'm giving it. We need to understand what gene therapy is, why it's important, how it could look for HIV. We need to start conversations with patients to understand what they want, because it would suck to spend years and billions of dollars developing a therapy that it turns out patients aren't interested in because it doesn't actually meet their needs. We have to have the ability to have clinics that are ready to implement these kinds of therapies. All of the gene therapy trials that have happened to date have started primarily in academic research centers with professors like me and others who are on this call, Dr. Steve Deeks, Dr. Hans-Peter Kiem, others who have brought these to the clinic using facilities like the one in the picture I showed you earlier at Fred Hutch. And that is the way that a lot of local patients will also get access to these kinds of trials. And so we need to enable academic centers to be able to start clinical trials and clinical research into these kinds of therapies. I think about it like, I'm not the smartest person in any room. Collectively, more minds you have thinking about the same problem, the more likely you are to get it to an affordable and sustainable solution. And so the more people we can get thinking about this, the more ideas are going to come that will allow us to get to the place where we actually have a useful, beneficial, safe, effective therapeutic that's going to be affordable and sustainable. Now, I just talked about new ideas. We need new technology development. We have to have new ideas. The stuff that we have so far, not all of it's going to parse out, and we need more ideas coming to the table in order for us to get to that goal. And we need a governance infrastructure in place to manage all of it. We have to have public trust in the new therapies that we develop. Now, in the US and in Europe, we're lucky to have the FDA and the EMA, which have already been through approvals of gene therapies, but we haven't yet gone through that process with the African Medicines Agency or any country in sub-Saharan Africa. And each needs to be able to enable not just the basic research at the academic level to do gene therapies, but also the clinical trial work and the oversight to ensure the safety and the risk to benefit ratio is balanced, and the public can have trust in that process so that they'll accept a therapy at the end. It also means that we enable commercial development so that local economies get the benefit. It shouldn't be that an African country is doing all of the work to benefit a company in the United States that developed a therapy that was never tested on Africans. It ought to be that an African company should be able to develop that and reap the economic benefit. Important to that is our International Community Advisory Board, or our ICAB. And Michael Luella and Linda D, who are on this call, are both parts of that. You may also know Moses Supercharger and Jess Shee. As was mentioned at the beginning, we're working on both sickle cell and HIV simultaneously. So we also have patients and advocates in the sickle cell disease space who are part of this work. And I'll just give a quick plug for one of our GGTI members, Dr. Mohammed Draz. One of the things that came up earlier when we discussed with various patient groups, and specifically people living with HIV, this idea about we need you to stop your art so that we know whether or not the gene therapy is working, that freaked a lot of people out. They're like, all I've been told is stay on my art, stay on my art. Like, I don't want my virus to rebound. And what happens if I have a partner and I want to know if we can, have intimate moment tonight, and what do I do? And so one of the things that we've been working on is this idea of at-home viral load testing. Something that, like a diabetes test, you would take a drop of blood from your finger, put it into a cassette, only in this case, you don't need a special device to read it. You can actually use your cell phone camera to take that picture. And Dr. Draws, who's at Case Western Reserve University in Cleveland, is actually developing a prototype device for this that we're hoping can be implemented into some of our clinical trials in the US, and then hopefully commercialized after that. So bringing these types of treatments is not just about getting the clinical parameters together, but also about bringing new technologies that make the quality of life better for people living with HIV. Where do we go from here? Well, taking gene therapy inside the body would be the most transportable, making it like a vaccine, essentially. You go to the clinic, you get your injection in the arm, and then hopefully your treatment is completed. That would really be the ultimate goal. Some questions we have to answer in that are, can we get enough gene therapy to the cells that need it for that to be therapeutically beneficial? And we need to make sure that if the gene therapy happens to get into other cells of the body, that it's not going to cause problems. And those are two questions that lots of groups are working on. I think you're going to hear about some of those during the webinar over the next few months. And so I'll just tie this up so we have a few minutes for questions. These are all the different lectures that this group has been so awesome to put together. And I hope I've given you enough of an introduction that you ought to be able to understand it. If you want to do some more work on your own, I also recommend the American Society of Gene and Cell Therapy's Gene Therapy 101 Patient Education Resource, just at asgct.org, shown in the upper left-hand corner here. And with that, I will just say, what questions do you have?
That was really great, Jen. Now, you know, we've only really had a couple of themes. One of them initially was, I think, really more of a question about off-target issues. And the others have been answered in the chat, except that maybe we should let Siggy, oh, there's a poll there. Anyway.
Yeah, I
we
put
should
that
let,
up for people while we're talking and we're wrapping up.
okay,
Just
great,
a quick
Michael.
evaluation.
Yeah, great. I'm glad you got to it. In other words, I just sent him this 2 minutes ago. But anyhow, we have the off-target issues, and Siggy had a lot of points and questions that he made. So in the time that we have left, if you could address off-target issues just quickly, and then we'll see if Siggy has anything that he wants to say or any questions he wants to ask.
Yeah, so in terms of off target, we kind of think about it two ways. So the first is off target in the blueprint. Like I said, you know, sometimes we're trying to turn off a gene like CCR5. It's a very specific place in the blueprint, and that's the only place where we want the change to happen. Off target could mean that we make the change in CCR5, we shut it off, but maybe we also accidentally hit some other gene over here. If that gene is not important to a T cell, that might be okay. But if it is important to a T cell, we could have other problems. So we think about off target in terms of in the blueprint. We also think about off target in terms of the cells in the body. So what happens if we accidentally turn off CCR5 in the liver and not just the T cells? Well, this is where each situation, each gene therapy is going to be unique. And it's an important question to ask every time you see a new approach. In the example that I gave of CCR5, we don't think that's a problem, but that's because we know that people are born with this mutation. And when they are born with it, it's in every cell of their body, not just their T cells. And they still live to be adults with a normal lifespan and a pretty standard health and quality of life. So we don't think that that's going to cause a problem in the liver. But when you talk about adding a new gene to this, like a CAR gene, you could say, okay, it really only matters in T cells. T cells are the only cells that can do anything with a CAR gene. But is there a problem if a liver cell expresses a CAR gene? We don't know. We don't think so, but we won't know until we look at that. Now, in terms of latent cells, where we go in and we try to turn off the HIV genes so that they're not very good at making new virus, we're trying to get into lots of different kinds of cells. I showed that slide, all the different cells that can become latently infected. We have to think about, well, we want it to go into lots of different cell types, but what happens if it gets into a cell and there are no HIV genes to turn off? What will it do? Chances are nothing. That's the way we try to design the system, but we won't know until we try. So in terms of off-target effects, they're definitely a question that I encourage you to ask every time, because it will be different for each approach.
There was an interesting question that someone asked. They were wanting to know if it's legal in the US to do human gene editing, or is it just in animals at this point?
I think she got two answers though. It's like 1 minute of.
Yeah,
so
yep.
She did get two answers, Christine.
Yep, and the answer is there are two kinds of gene editing. When we talk about like the legality, you'll hear these two words, somatic and germline. And those are scientific terms that mean like liver, eye, skin, T cells in a person versus that's the somatic types. They've already decided what they're going to be and which parts of the blueprint they're going to use, versus germline, which would be like an embryo or a sperm or an egg. And germline is not okay to do right now. There's been a moratorium issued by the World Health Organization because we don't know what the potential issues over generations could be, but somatic is allowed.
Okay, because we did have a doctor come in, do a presentation, and he had done it on mice with human DNA in the mice, and it was successful. It was successful. They did become not positive, but he said we're far off from that happening for humans at this point. So I was just curious how this is going to work, and you know, to get us to the point where you can edit my gene, get rid of my HIV, and then it's all, you know, for the rest of my life, no more art therapy, no more aging with HIV, all that stuff. That's all. But thank you.
And it does. You're welcome.
Wonderful
It does take a long time.
Siggy, did you want to have any comments that you wanted to make? I guess that means no.
And I just wanted to point out to everyone that we still have, I put a poll up and a little evaluation. We're just asking what you liked about today's presentation, what you think could be improved, and what other questions you may still have. If you have a chance to fill that out before you sign off, that'd be great. I'll keep that up for a while still for people who might be filling it out.
Yeah, I have a longer one too. I thought my assistant would get to it, but somebody got into our bank account and he's getting over COVID, so I don't have a link for it. But we can send you.
Linda, do you want me to post the link that people can download?
That would be great. Because I'm trying. I see this link that you just sent me, Richard, that I asked you to do, but I wasn't sure if that's, you know, I tried to open it and it didn't open to what I sent. So does this link work, Richard?
Fingers crossed. It just rather than being able to fill it out online, you just need to download the Word document and then complete it and email it to us if anybody
that
has
would be
the time
great
to do that.
That's quick. We'll talk about quick work. So it's just a one pager that gives us a little bit of information about you. You know, I mean, fill in things that you feel comfortable with. If you don't feel comfortable telling us how old you are, don't tell us. But if you also, it really gives you a couple of questions to kind of express what you thought about today's call, which would be very helpful to us. So if you wouldn't mind doing that, or if you just want to do Michael's 3-question evaluation, that'd be great too.
And also notice that Jen did post into the chat for people to download the article. If you wanted to take a look, she put that up there. I wanted to make sure everyone got to see that.
What about that gene therapy 101 that you were talking about, Jen? It was on one of your last slide, or you're next to the last slide.
Yep, I will pull it up right now and I'll put a link in the chat for everybody.
Yeah, because I think that might be really, you know, it's always hard for us because we just don't know whether we're getting down enough for people to understand this stuff, or whether people understand everything we're saying. So if you could help us understand, you know, if you understood what we were talking about today, that would be very helpful.
Yeah, and in particular, if there were parts of it that didn't make sense to you, that would also be awesome to know, because it gives
Yeah.
us some ideas on how to change or things we need to try different approaches with.
Right.
Mm.
And thank you so much, Michael and Jen. I thought it was great. I think you thought it did a great, very comprehensive job.
Yes. And I want to
Anybody
thank-
else want to say anything before we close it up? Remember, we have another call next week where we're going to be talking about CRISPR. That is going to be extremely interesting. So we're going to actually start to get into different gene therapy interventions. So please join us.
Right.
And then we'll go from there. And maybe we'll send the evaluation also afterwards. Okay?
Can I just say it was very well done. The presentation was great. And so you're saying the next one's going to be for CRISPR. So I actually get to understand what the cluster,
Clustered, regularly interspaced
regularly
repeats.
Well, we hope you'll have a better understanding.
Halotraumatic repeat. I know what it means, but I really need to know more about that.
Well, we have next week, we have the guy that really pretty much is the father of CRISPR. So he should be able to tell us. And we also have somebody else that works with him at, at one of the other MDC sites. It's actually called CRISPR for Cure.
Yeah.
So hopefully you will
there's quite
join
a few.
us and you'll have a lot of questions and hopefully they can answer for you next week. Same time, same station.
Yes, thank you. That's awesome. I've
Great.
been hearing a lot about it. And Germany, there's some woman in Germany doing some really good things with
Well, in a
CRISPR.
couple of months, we're going to have a doctor from Germany who's a male, who's a big deal in the same arena. So
Oh, okay.
yeah,
That's
we
going
try to
be-
cover everything.
Stay tuned. Yes. Very
Yeah,
awesome.
stay tuned, please.
Well, thank you so much, everyone.
All
Thank
right.
you. All righty, bye. Thanks again, Jen and
Bye-bye.
Michael.
Yes.
Bye. Thank you.
Mhm.
All right. Let me do that.
So it looks like people are dropping off. Michael, can we send that other evaluation to people, the link? Or I
Yeah.
can, since we have more time, I can get a link made maybe. Or maybe you can make one for it.
I was just
for
going to
me.
use the one that Richard used.
I didn't, well, I tried it just to open it and I didn't see where it opened up right to it.
It didn't what?
It didn't open up right
It opened up for me.
did it?
Yeah.
Okay, if it opened up for you, then just use that.
Yeah, that's what I was going to do.
Perfect.
All right. Cool.
Speedy Gonzalez,
All right.
Richard.
I'm going to
Okay.
stop. Yep. I'm going to stop the webinar. Thanks, Jen. It was great.
Thank
It was
you,
a lot to
guys.
cover, but you did it well, expertly.
Really excellent, Jen. Thank you so much.
Yeah.
Thanks,
Yeah.
guys.
And next
Bye.
time, Richard will be paired up with, actually, CRISPR is not yet. It's a third
It's
meeting.
right. I was going to say it's Jim Riley next week.
But we
Right?
have Jim Riley, who I just sent an email to last night. So
Cool.
I'm just a little fried.
right.
Anyway,
I'm going to end this.
okay, thank you.
Ciao.
Bye.